In this exercise, we're looking at an example of soil microbiology. Sometimes, for various reasons, you may want to know what the level of microbial growth is in the soil sample. And so there's several different ways that you can do this. I actually know someone who developed a method for using fluorescent microscopy where she takes the soil. She goes through this process where she adds a fluorescent dye. She uses two different dyes. One will bind to bacteria, one binds to fungi. And then she looks at it under the microscope. And basically, if it's glowing one color, it's bacteria. If it's glowing a different color, it's fungi. And these will only bind to living cells. And so she usually takes the sample, dilutes it, puts it on a special type of slide that has a grid similar to what we use for blood counting. And so she can actually sit there and count the number of little glowing specks that she sees, and it's glowing because of the fluorescent dye that is attached to the cells. And then she can go back using the dilution factor and calculate there's so many bacteria per gram or so many fungi per gram of soil. Sometimes you just need to get a rough idea. And if you don't know or have the means to do the fluorescent microscopy, probably the most common way is to do your standard plate count. And once again, you're going to do a serial dilution first so that you can get it within that range when you plate it of 30 to 300 colonies. And then you relate that number to the dilution factor to get back to determine how many bacteria per gram of soil. As technology improves, we always get new methods and procedures to use, and so some people can actually test for the presence of DNA in the soil to give them an idea of the concentration or the load of microorganisms from that method as well. So for our lab, we rely on the standard plate count, and so in this experiment, what was done is they had a soil sample, we took it, we did a serial dilution, and then plated it out on the plates. And you'll notice that at the highest concentrated version of the sample, you have really high extensive growth here. I believe we used potting soil in this, so you would expect, it wasn't sterile, so you would expect to see a lot of growth. That's too many to count. We have a lot of growth here. This is still too many to count. And then you see the amount is, with each successive dilution, is decreasing, which is what it's supposed to do for a serial dilution. So you get down in the range. You can see two very large colonies here, but there are several smaller ones in here. This was done using a pour plate, and so with a pour plate, remember, you also have colonies not solely on the surface, but also embedded in the agar as well. But you get the count, you relate it to the dilution factor, and that relates it back to the original sample, so that ultimately you're able to say there is X number of bacteria per gram of soil.